People, they say that they have all the, they say that the West or the advanced countries have all the resources available, and they do not want to share with the poor countries and other scientists or other institutions. It's not true. With NIH, what NIH is getting out of it? Nothing. They know that all the institutions in USA or in Western countries and North America, they are all rich countries. They can buy the software, right? So basically, they are doing a service to us, to underdeveloped countries. The countries who cannot buy these software, but you can use this software to do your own research. So the NIH funded this research, and it is the duty of these scientists who are working on it. They keep it updated. So since I'm teaching this software, I didn't find any problem. So they just have all the updates available, and it's a very good software. For density maps. I don't know if some of you already have done NMR or something. So where we have the density maps of the structure of the atoms, you can use it for sequence alignment, for docking results, conformational ensembles. You can make movies also using this Chimera software. It's such a good software. And I cannot teach, unfortunately, everything that you can do with this software. I have only a couple of things that I mentioned related to the molecular modeling so that you get an idea. The first Swiss model, and I'm sure, again, Swiss model, you can do tons of things. I'm showing only that how can you use it for structure building and how does it work actually. So here, this is your sequence, the one that I gave you, the unknown sequence. You get it from your professor or something that you just pick up from the available sequence databases, and you said, I want to work on this. When you have for example, if you are a faculty member and you want to have a project for your students, you can ask them to pick up this sequence from some of the databases available, which have a record of all these protein sequences that we don't know the structure and function yet. So just pick up any sequence from there as your master project or PhD project or MPhil project. And then what actually the server does, we just copy the sequence there and then we press hit run. And then we are waiting for results to be displayed. But in the background, the server is busy doing all this. So what it does from your sequence is try to recognize a template sequence in already existing available databases. And from there, it tries to do the alignment, sequence alignment with those databases to find out whether it is finding the right sequence, the right template or not. And while it is doing it, it starts also building the structure of your protein. The first, it makes the backbone of your sequence that you have mentioned, and if it has any loops, hanging here and there, they try to model the structure of those loops, then side chain modeling, and once it has determined a template, a structure for your protein, then it does the model optimization and validation. Have you heard about Ramachandran plot? Yes. In NMR, perhaps you have heard about. What is Ramachandran plot? Ramachandran plot? You guys are nodding your head. What is it? You don't know Ramachandran plot? Have you heard about it? Anybody who's from biochemistry background, I know that in the book, Stryer book, we all learned about Ramachandran plot. That Ramachandran plot is very, very important. Yes, what is it?
It tells about the secondary structures that exist in the proteins, like alpha helix or beta sheets or psi and phi angles.
Psi and phi angles? What psi and phi angles? What actually, what is the purpose of Ramachandran plot?
It gives a possibility that where these what we can say, phi and psi angles can exist, and the best possible structure of the protein.
What do you mean by best possible structure?
Because there are many.
So when I asked you to define what is the best possible structure, is that you are still stuck on ppm? Did you find something about ppm? Yeah. Did you search in ChatGPT?
Yeah.
And you get something or no? You get something? Okay. So when you go today, then just write Ramachandran plot. And then Ramachandran plot, basically, anybody who... No? Give up? You just have your NMR exam, guys. The Ramachandran plot is the guy, as the name suggests, Ramachandran. And what he did, he tried to, there are different angles exist in the protein structure. Phi angle is between which carbon or nitrogen?
Carbon and nitrogen.
So your amino acids, they have a parallel skeletal structure, and where the peptide bond connects the carboxyl group of one amino acid with the amino group of the another amino acid. And the bond in between is called peptide bond, right? So this peptide bond can be rotated. And when this peptide bond is rotated, how the structure is changed, right? So the proteins are continuously breathing in, breathing out. They're continuously rotating. The structure of those atoms around those angles or the bonds continuously in fluctuation, right? But now, how much they can fluctuate around that angle? If we fluctuate so much, the atoms will start colliding with each other, right? And it will destabilize the protein structure. So what Ramachandran did, and it was a huge thing at that time, because when he found out this, when he drew this Ramachandran plot, there were no computer existed at this time. There was no AI, there was no algorithm. Right now it seems very easy. You can make tons of such Ramachandran plot yourself. But at that time, this guy did everything manually. And manually he find out how much you can rotate each bond or each angle in any given amino acid, in any given protein structure, right? He calculated it and he let us know that if you go beyond certain number of the angle, certain, if you twist the angle beyond certain wavelength or so, they will start colliding with each other and it will destabilize the structure. So this Ramachandran plot, you didn't see all this yellow thingy here? The yellow ones are the one where you can rotate your phi and psi angle and there will be no clash between the atoms. But the white region, if your angles, you rotate them so much that if they fall in the white region, the atoms will start colliding with each other. So this Ramachandran plot is actually the basis of the molecular docking. So this information, we have fed this information into the docking softwares. In docking softwares, when they're trying to dock our drug against the protein, our protein of interest, the docking software is actually trying to put the compound everywhere, here and there, while at the same time it is trying to make sure there is no clash between the atoms. And based on that, it tells us, that your compound best suited in this position, in this conformation, right? And if you try to rotate it a little bit here and there will be a clash, it will destabilize either the drug compound or the protein structure. That is the basics, very basis of the molecular docking softwares, right? When we are busy, we will just give the command run docking and waiting for the results. Computer in the background, it's just busy doing. So this optimization is done through the Ramachandran plot, right? So the server is busy doing all this. Here I just make it very simple schematic, and if you complicate it, then this actually is happening in the background: sequence alignment, model validation, optimization, looking at different bond angles, and so looking at the spectra, and then finally giving you the final refined structure of your protein. So based on all the background information, how your protein structure should look like. So far, so good? Any question? Okay, so that was the background about the homology model. It's this model server. How does it work? How you can use it? Just go to this link, write it on a new tab, and then a page like this will show up. In this box over there, you can copy your amino acid sequence. So I copied my amino acid sequence in here. And some of you, if you are familiar with, I'm not going into too much detail about the FASTA sequence. If you have access to FASTA sequence, you can also copy the FASTA sequence. If not, forget it. Then just use your protein amino acid sequence, copy it over there, and then click on the search for template option, right? The search for template option in here. So what are you saying? You are just giving the sequence to the server and asking just look for templates which are similar to the amino acid, and then let me know the structure, how the structure looks like, right? In just one single command, you will get, it will tell you that these are the proteins I find which are giving a structure similar to your protein of interest. If you look at this sequence, it's a very tiny protein. Why is it tiny? As compared to huge proteins and structures that we have seen. For example, I show you, there is a huge structure. For example, this one has a huge structure, this is a tiny one. So my question is, why is it tiny?
For the short sequence of amino acids.
Because it's just showing the structure of your 50 amino acids. See how much powerful the program is? Only your 50 amino acids. Just telling you based on your 50 amino acid sequence, your structure should look like this. But it is not just based on 50 amino acid sequence. It has compared these sequences with the existing template structures, and based on those structures is telling you your amino acid sequence should look like this. If I ask you manually draw the structure of these 50 amino acid sequence, you will draw it differently because the only information you will be using those 50 amino acids in front of you. But this database is using all the background template structures and based on that optimizing and telling you this is the best possible structure for your 50 amino acids, right? So here, not only this, it is giving you other options. You said, okay, you know what, according to this, your protein should look like this, but there are other options available. If you want, I can make a structure based on this protein, or based on this protein, based on this protein, and based on this protein. So depending on whatever information you have available, if you want to check with other protein, and just click on it and see how the structure looks like, right? But whenever you're working on a project, always try to have as much information as possible about your protein, whatever you have available. We already have used other databases. From those databases also we have learned something about our protein of interest. So based on that, maybe perhaps you say, no, I don't want to use this one, I want to use this one protein. And I want you to design the template based on that. So you click here and then click on the model results. So when you're done, something like this will display here, right? So for me, for example, the structure almost look the same because I agreed with what the server suggested to me. So here you can also have much more information other than that. It is telling you that your 50 amino acid sequence and the template structure that you have chosen or the system has chosen for you, there is around 86% sequence identity in those structures, right? And also it's telling you the amino acid sequence, and here, for example, it is telling you that how your sequence is different with the template structure and where it is different. For example, here in your model, G which is guanine, right? This guanine, the glycine is replaced by, in the template is replaced by A, which is alanine, right? And so on, like other places. So exactly you know that at what places your structure is different than the existing one, right? And there are many things you can learn about it, just everything which is clickable, click on it and then go and just learn as much as you can. What I would suggest, I'm asking you here, because you need the PDB format structure of this protein, so you can download it here, click on it and download it. This file will be using for the docking studies, right? So just keep that file safe somewhere and then we can use it for docking. And there are some questions I want you to answer as you move on, and the answers are usually here. And if not, then just click on, for example, structure assessment and other things, you will get more information. If you click on structure assessment, you get more information. As I said, wherever you find a clickable a link, click on it, and then, for example, here, it is telling you, okay, I have made the structure based on Ramachandran plot. So this is the Ramachandran plot related to your protein of interest, right? But it is telling you at the same time, there are certain bond angles which I'm not happy about. Something wrong with those bond angles. They're bad angles, right? Then they say the actual generally that how your structure is strong and how it is weak, and actually you can modify and change the orientation around these structures to see if you can stabilize it, right? So here, for example, it is showing you out of 515 angles, there are 3 angles which are bad. And which are those angles? Where are those are located? Number 1, number 2, and number 3. And also highlighted in here, right? One of them is I highlighted by myself. Right? So here you can get more information about the stability of your protein and how much good the structure is. That was all about server, this Swiss model server, right? But this is not the only thing you can find out, because by far today's lecture is about only structure and docking studies. I'm just talking about the structure, but look at the power of that model and you can do much more there. As I said, unfortunately, I cannot teach everything in the class, but if you're working on a project, for your students, if you're thinking about designing a project, this is the best starting point, right? Okay, Chimera, okay, now come to the Chimera. Chimera is a software that you'll have to install. And again, it doesn't take too much space, easy to install, doesn't take too much time to install. For the Windows, this is the site you should go and for the Apple, this one. Actually, how many students are using Windows here, Microsoft Windows? And what about Apple or the Mac?
I changed. I used to. I'm yet to learn on Windows. I use Mac, but it's every time you have to pay or go to Apple Store.
The problem is because majority of the software that we have available, they are not Apple compatible. Unfortunately, I don't know.
Yeah,
but still, I find people who actually who use the Apple computer, they love to use Apple computer. But why for these type of work?
It's nice.
The graphics are very good in Apple computer.
It's quick.
The graphic card is very good. But unfortunately, I always have problem. Whenever I'm having students and I ask them to download this into software, usually the Apple students, they come in, they say, man, it's not working, I'm trying to install, especially docking for them doesn't work. Though Autodoc Vina, the one I will be using, they have a version available for Apple computer, but sometimes it doesn't work.
You have to update every time. You have to update
For the Apple,
yeah.
So there's something, some compatibility issues, but still people who love working with it, they still love. If they are okay with all the software they are working with available for them, then they are fine, they use Apple. But I find with Windows you have less problem. For me as instructor also, because they still come to me all the time. And sometimes I have solution for them and sometimes I don't. I say maybe just go and explore on Google. Here, for example, they are making it available for both Windows and Apple. Chimera should work. I never had anybody having any issue with Chimera, even with Apple computers. Okay. Now the Chimera is a really powerful software, as I mentioned before, and it's very easy to use. Not difficult at all. Even if I don't teach you anything, you just go and explore these tools. These are the tools available. This is the toolbar, right? And this is the one mostly I use, action, or here you go to tools. Just go and explore, click on everything and see how does it change the structure, function, sequence, mutation. You can make a movie out of it. So many things you can, just explore by just going to these tools and then see what it, actually telling you about. So once you open the Chimera, UCSF Chimera, and the very first thing I'm asking you to fetch the PDB ID of your target protein. So far you have learned what is your target protein, which protein your protein sequence is actually closely matched to. That is actually your target protein that you'll be isolating from, fetching from PDB. So what I did here, this is my protein from PDB database, right, with which my sequence closely match, and this is my 50 amino acid sequence, right? So now you can, real time, you can see how close your 50 amino acid sequence with the target protein is. And for this, initially you were just relying on the database, whatever information they were giving. You were just relying on the Swiss model, right? Here you are just checking it by yourself. So what I did here, I have this my protein PDB ID is 1E9Z. Use your protein ID, and here's the amino acid sequence. I'm trying to see how much they are overlapping with each other. What is the structure and the sequence comparison? So for a structural comparison, you just follow these go to tools like here, then go to structure comparison, then matchmaker, and then, for example, here, go to tools, structural comparison, matchmaker. When you click on it, a pop-up like this will open. So it is asking you what you want to match. So here, what I'm telling you in the one reference section, I'm highlighting this protein, and another section I'm highlighting this protein. So I'm just telling them compare these two with each other. So once you're done clicking apply, and then here you get the comparison. So here the entire protein, the PDB, that you get the PDB structure for the entire protein, and these are your 50 amino acids. So see, now here you can see in the real time where your 50 amino acids is superimposed on the actual native protein structure, right? So this is one thing, and also now you want to see how much the sequence similar to each other. For this, you just go to tools, structural comparison, match, align, and then a pop-up like this will open in here. So here you will have, this is the PDB ID from the entire protein, and this is the Swiss model generated structure. So this is the sequence on the top is from Swiss model. This one is structure here, and this is from the PDB ID, and you can see exactly where the amino acid sequence is similar and different. So if you click on this here, info, so it will show you the percent identity, which is I don't know how much, if you can read it, it's I think around 86%. It's showing 86% comparison, identity between my 50 amino acids and this protein. 86% means it's not 100% identical, right? It looks like my sequence was taken from a protein like this, but not exactly from there. It might be a very good opportunity, perhaps it's a novel protein that you have discovered. You have find out a novel protein, you know its structure, you know its sequence, and you know what it is similar to. So it is similar to a big protein, like this is how the COVID vaccine was actually generated. In 1.5 years, otherwise it takes 9 to 10 years to come up with a drug or so. So what they did initially, they find out a small tiny sequence, right, of the virus, and then from there they try to find out which is the big, huge protein it's similar to. And because they knew all the drugs available against this protein, based on that, the same drug component they tested against the COVID virus or protein that they discovered. And this is how it became easy just in 1.5 years. Usually, in fact, it's 1 year they were able to have a vaccine against COVID. To see all this available information, how it was used to come up with something so fast and save so many lives, right? So this is for the structural comparison. You can superimpose the structure, similarly sequence comparison. Okay, this is a fun exercise. You don't have to answer any questions. I don't have any questions in here, but just to give you an idea that what other things you can do using Chimera. So what I did, I'm trying to compare the structure of myoglobin with hemoglobin. Myoglobin is 1 chain and hemoglobin with 4 chains. And here what I did, the PDB ID for hemoglobin is 4-HHP and for myoglobin is 1 MBO, right? So here I have hemoglobin protein consisting of 4 chains. This color is not showing up actually, but there's a chain also. The chain A, B, C and D. This is hemoglobin. And here what you're seeing, the red, the magenta color superimposed on the red line, right? So the magenta one is from myoglobin, is the chain A of myoglobin, and the red one is the chain D of hemoglobin. So when I try, because I've heard many times that the myoglobin and hemoglobin, they are superimposed on each other, the 3D structure is same. Now I'm trying to see whether they're actually same or not. So here using these commands, if you just follow the commands, I've been able to superimpose the myoglobin protein chain on one of the hemoglobin chain. And again, here I'm doing a comparison that how much they are close in structure. So if you look at the CTF, what is CTF? CTF? C-terminal fragment of both proteins, the protein D, chain D of hemoglobin and the chain A of myoglobin. Here I see a little bit difference, right? That they are not 100% similar. The 3D structure of myoglobin and the 3D structure of one of the hemoglobin chain, they're not 100% identical. And where are the differences? I can see myself. So here at the CTF fragment they are different, and at one of the loops they are not exactly superimposed on each other. As compared, when you look at the heme group, you know that both of them they contain a heme group. Myoglobin and hemoglobin. What is a heme group?
Iron containing.
Iron containing? 4 porphyrin rings, and it's a prosthetic group for a protein. A prosthetic group is a non-protein component of any protein. So for hemoglobin, it has iron. That is the reason it is able to absorb oxygen and then transport it from lungs to the tissue. Similarly, for myoglobin, it has a heme group consisting of an iron atom. So the heme group in both the proteins is highly conserved. It's highly conserved. So and we can see it here. So this is the heme, it looks like one thing. The heme group from myoglobin and heme group from hemoglobin, they are here exactly superimposed on each other. This shows that evolutionarily these heme groups they are related to each other when the proteins were evolved gradually, right? The heme groups usually is preserved in most of the proteins which contain the heme group. Okay, so this is the hemoglobin and the myoglobin. You can yourself. So here I'm giving the example that we already know, right? But again, as I said, for your own project, you can come up with 2 proteins that have never been reported, and you are doing a comparison. And here, how you can do the comparison. I can write a complete manuscript based on that. Just for the structural determination, how they are different. Take different pictures from different angles, rotate it, and maybe I find CTF and loop difference. When you animate it, perhaps you find more differences. And then try to correlate that difference with the sequences, right? And then one complete paper manuscript is ready for you to get published. Right, and here is if you want to compare the amino acid sequence, you have to choose which the myoglobin chain you want to compare with which chain of the hemoglobin. I chose chain D. You can choose chain C also, chain B or chain A, and then do the comparison one by one. Again, here is the comparison. It's around only 24% identity. The myoglobin chain and the hemoglobin chain only 24% identical. But 3D structure is almost 86% overlapped. right? We need to find out the reason for that. How is it possible? When we say the sequence is similar to structure and the structure then is similar to function, now the sequence similarity is only 24%. How the structures are similar? So it's a question. It's a scientific question, and you can design a master thesis to answer that question. Design a project for your student, for yourself. Write a paper on it. That how 24% sequence identity leads to 86% 3D structure identity, right? And then how the structures, when the sequence is so different, their structure and functions are similar. On the other hand, we propagate that sequence leads to structure, same structure, and the structure leads to function. So why is it different in here, right? You see, there's so many questions, scientific questions that we have no answer about. So if you are very creative in your mind, every time you read something, every time you do something, you find out a new question that you can find an answer for. And you can design a new project with it, right? Develop your skill sets in this, offer these solutions to the scientists, market yourself, and then here you have your own startup company doing this. Okay? Which vehicle is spelled the same forwards and backwards? Any idea? Any idea about the cars?
Race car.
Race car? You have a race car? No? A toy race car? Anybody has a toy race car?
I had it.
You had the actual one? Actual race car?
No, actual one.
Do you agree what she said? Maybe I think people didn't hear. Which vehicle is spelled the same forward and backward? Any idea? I'm looking at boys because usually boys are good at. What is the name of the vehicle that is spelled backward and forward the same spelling? Train? Train, no. Train and MAIRT, no. Different. Sorry? Who's saying something? Rickshaw? How come? Rickshaw is RICKSHAW. The WASKICR something, right? Huh? We don't know? You see this? Deepak?
Race car.
Huh? Isma already mentioned it. When I asked the question, she immediately gave the answer. The answer is race car. You see race car backward and forward same spelling RACECAR, RACECAR. Race car. Okay, next. Come to the boring lecture. Time docking. This is the last step for today's class. How would you do the docking? Any idea about docking? Why we use the word docking? What is the meaning of docking?
When ships are docked.
Who is this speaking? Oh, yes.
When ships are anchored near the port, so that is.
It's called docking, right? The ship has been docked, right?
Yes.
But then why we use this terminology, same terminology for drug designing or something?
Because compound or ligand is anchoring into the active site of the protein, so that's why we are.
But it doesn't need to be active site all the time.
Yeah.
Sometimes it's just on the binding site, right? And sometimes it's a site other than the active site, and to activate the protein, also inhibit the protein. So yes, this is the literal meaning of docking, that you're trying to dock a compound on the protein, right? And basically, if you want to know what the docking is, it's basically matchmaking, right? The venue is computer, and the purpose of it just to predict the druggability of any compound or any line. But docking cannot be used only for drug design, right? Docking could be protein-protein interaction, could be ligand-ligand interaction. It's not always about docking a compound on a protein, right? It could be anything. You can use docking to find out how two proteins are interacting with each other. Why protein interactions become important? Why we want to know about it?
Because you can design a new compound from this interaction. And new structure of the proteins that from different.
So you're right. But basically, if you are a biochemist and you worked on those PCA cycles, protein metabolism cycle, lipid cycles, the glucose cycle, how the glycolysis is happening, right? So the different set of enzymes are coming in action to metabolize single glucose molecule, for example, right? Those enzymes are interacting with each other. Right? So if you are working on interaction and proteins interaction with each other, you're working on deciphering an entire pathway of metabolism, for example, for protein, for glucose, for cholesterol, for lipids, or for nucleic acids, right? So the protein interaction becomes very important. It helps you to find out the basic, fundamental basics of biochemistry, physiology, and anatomy. Protein interaction is also very important. It's not only the drug and the protein interaction. There could be ligand-protein interaction, ligand-ligand interaction, and the protein-protein interaction. And how we do it? So we need to have 3D structure of both of them, right? We can have the 3D structure of only one of them, but it's easy that if you have 3D structure of both of the proteins or the ligands or the ligand and protein is known, then it's easy to do the docking experiments. And what actually is happening here? So this is your protein, right? And the compound is already bound to it. And this is your compound, and this is the active site or the binding site of the protein. You see how the structure of the ligand is complementary to the active site or to the binding site, right? Complementary means you look at the structure, how tightly it is fitting into that structure, the active site or binding. It's called the complementarity of the structure. It's very important. So there are 3 things that come in action in a compound, a protein, when 2 different molecules are trying to interact with each other. Geometrically, they have to fit in. Geometrically means complementarity between the structures. Electronically, they have to complement each other. Electronically means if there is a negative charge here on the ligand and there is a positive charge here on the protein, they will interact with each other and they will keep it in place, right? The binding over there will be tight. And energetically means how much energy you need to maintain this structure. If the system needs huge amount of energy to maintain the drug and the protein in this conformation, what do you think? It's a good thing or a bad thing? Because it means that the structure will not be stable, right? And you need huge amount of energy, which is not possible. Electronically, energetically, the entropy of the system will be high or low? High. The orderness of the system will be disturbed, right? So which is, in nature, we have highly stable protein structures. We have very high stable lipid or nucleic acid structures, right? So it has to be energetically very favorable. Okay, now I want somebody to do this calculation. So remember the Ramachandran plot I mentioned, and this is the basic of all the docking algorithm and the software that we have available. Ramachandran, what he did, he told us, these are the phi and psi angles. If he's saying if you rotate these angles up to this range, for example, here is beyond minus 80 something in here, right? So if you're maybe around minus 60, so if you're rotating phi angle up to minus 60 and this angle is here minus 30 or so, and it is falling in this region, the structure is stable. But if you're rotating it up to extreme range and your final structure is falling in the white region, the structure is not stable. That's the basics of all the docking algorithm. That information has been fed in this docking algorithm. And how they do it further, so this is the information they feed. And also you need to know the van der Waals forces between the atoms. You need to know the electrostatic interactions, hydrogen bonding, desolvation energy. You have idea of all these things? Yes. What are the van der Waals forces?
Intermolecular forces.
Van der Waals forces, usually when the atoms are far, they are ineffective. They become effective and they become questionable or what when the atoms come close to each other. When they come close to each other, there is chances of having static crash in between. So this is called van der Waals forces. The van der Waals forces are keeping them at distance from each other. We don't want them to collide with each other. It's called van der Waals forces. Electrostatic forces?
Positive.
Yes. Interaction between negative and positive charge is called electrostatic forces. What is the hydrogen bonding? Yes. So hydrogen bonding, the hydrogen atom should be a part of hydrogen bonding, right? It's a weak bond, it's a strong bond. Hydrogen bond is strong bond? No, it's is it a covalent bond or non-covalent bond? Non-covalent. It is one of the non-covalent bonds, which non-covalent bonds are not strong actually. But hydrogen bond is an exception. Despite being non-covalent bond, it is a strong bond. Right? And it is the main, and how can you, for example, give you the water, when you can see the water is making a stream of the continuous, molecules or water molecule connected with each other. That's why you're able to see the stream of water coming in. It's because of the hydrogen bonding. The cohesiveness of between the water molecules, the amount of water molecules that results in they pull each other. This is one of the reasons the water from the underground pulls all the nutrients through the roots, the stems to the leaves because of the cohesiveness of the water, and this cohesiveness is coming from hydrogen bonds, right? So hydrogen bonds, these type of covalent and non-covalent interactions, they determine the final stable structure of your protein in your drug, right? So all this information, I'm just giving you only very brief definition of those interactions. They have been placed into these docking softwares. So while you're at front, just press the button, run docking, waiting for the thing to happen. In the background, the system is busy doing all this calculation to give you a binding energy, which is the most stable for that particular pose of the protein and the compound. Now what I want you guys to do, so this is a formula for the van der Waals forces and hydrogen bonding and so on. So I will give you numbers.
Yeah.
Will you do the calculations?
Yes. No.
No?
Okay.
Who will do the calculation?
We will try.
You will try? If I give you the numbers right now? Okay, write down the number. You have the calculator? The phone? You can use the phone also. You know the phones that you have also has a calculator? Yes. So you can use that calculator. So if I give you the numbers and I just want you to give me the final answer. Would you be able to do it?
Yes.
No?
Madam, ये symbolic नहीं आएंगे.
Huh?
There will be no symbols like this.
Manually do it. Ramachandran did it many years ago. This guy, and there was even no calculators or so. He did everything manually. See how much hard those scientists worked at that time. Now you can do it very easily. Within a few minutes, you can do all the calculations. Okay, so I'm not giving you guys numbers. The majority of the students are saying that no, they don't want to do it, and especially you are saying it, you don't want to do it. Then you will ask for 10 minutes break, right? Okay, so that information, all these mathematical calculations have been fed in different algorithms, right? And it's not all algorithms have all kinds of calculations. Some of the algorithms are focused on certain calculations. Some of the algorithms are focused on a different calculation. Maybe one algorithm is just talking about only Ramachandran plot. The other algorithm is Ramachandran plot plus something else. Third algorithm may be just not talking about Ramachandran plot at all, and just using new set of calculations, right? So those they have been given different names. For example, Monte Carlo and the genetic algorithm, they basically use AutoDock as their main algorithm. So these are, and you know what the algorithm is basically? If I ask you to design an algorithm, would you be able to? Where is Dr. Riaz's student? He's hiding. You will be able to design the algorithm. Algorithm basically, you use Excel sheet, right? And Excel sheet, have you ever write any formula for any calculations?
Yes.
Like plus, minus, multiplication, division. The simple formulas you can make yourself, right? Everybody knows it?
Yes.
So this is a very basic of algorithm. The very basic algorithm where you can design a formula in the Excel sheet. Now just make it complicated million times and repetitive. Like you are doing exactly the same thing again and again, hundreds of times, and make it too complicated, too many formulas thing. So you don't want to do this manually anymore. So all that information has been put in the coding or the codes and the commands that we design for these algorithms. So these algorithms, for example, when you're trying to dock a protein and a compound, in the background, the algorithm is continuously running and checking the conformation of a protein and a drug structure in one orientation, then what is the conformation, all the calculation, then changing the angles a little bit and doing the calculation again, changing the angles and doing the calculation. They're just busy doing all this. There's all repetitive thing that they are doing at each conformation, a different conformation, right? So rather we do it ourselves manually, we are using the computers available and we're designing the algorithm, they are doing it for us. So this is how these algorithms are designed. So we'll be using AutoDock Vina. So whatever I'm talking about, and I think I mentioned yesterday, it doesn't mean it's all up in the air, imaginary things. The drugs have been designed using this computational model. And we have examples. So the first drug that we designed is 1995 as the carbonic anhydrase inhibitor. It is dorzolamide, and this is the structure of the drug. That drug came in the market just using computational talking, right? Later on, they did some clinical trial experiments. You have to, but initially, all the initial work was done through computationally. Similarly, we have imatinib for cancer, and we have tons of all those compounds. There are the drugs which are available in the market, and they came in the market using this primary knowledge that I just mentioned. While I was making the lecture, as I mentioned, I think in the very first class, I said, okay, let's just look at the startup companies who are working on doing docking. I find my initial search on these 9 companies. There are 9, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 companies, right? And then I said, okay, just communicate with any of the companies. I went to their website, Innovative Informatica, and usually they have some help desk available here on this site. So I start communicating with them, right? Many things. Then they're asking me that, I said, I want a docking done, how much do you charge? They asked me how many targets in ligand. I said, only 1 ligand and 1 protein target. And look at their response. I did not make it up. It is the response they wrote. Okay, since only 1 protein and ligand target is there, we can do it for you for our basic service charge of USD 1,000 plus 18%. USD 1,000 how much? How much Pakistani rupees?
2.8.
2.8? Lakh. 280,000 something, right?
Yes.
So this is what you have in rupees. And it is many, many years ago when I asked that question. Now the inflation has gone so much, right? Why don't you want to make this money? You want to make this money?
Yes.
Right? So just develop these skill sets. Develop these skill sets, come up with the creative ideas, broaden your horizon of knowledge, learn more about what is not known. There's so much that we don't know about many things, right? This is the reason millions of scientists all over the world are working, finding answers to those questions, right? So think about what is not known, and for this you have to be genuinely studying your field of expertise. Try to know what is not known, how can I develop these skill sets, and using this information I can contribute answers to the questions. And but at the same time, you're doing 2 things. You are disseminating the knowledge, plus you're making money, your own living with this. Right? This is what we do. That's the purpose of education. That we are going to the institutions, we are developing ourselves. We are learning skill sets. We are making ourselves valuable. So that when we've done our education, we can sell ourselves. So we are marketing ourselves. What I'm marketing over then, I'm marketing my brain, my knowledge, my information that I have gathered so far, right? You guys might be doing exactly the same thing. So you need to learn, beside education, the skill sets, and then once you are good at, sell it. That's what the purpose of going to these institutions. Other than learning and getting knowledge, and it's a part of knowledge. It's a part of disseminating the information that you have gathered so far, right? So this is only the basic thing, and then see how much money is it making. I know Zahid will come to me after the class, ma'am, can I get the link to this company? Do you want to do this? You are organic chemist basically, right? But and it has nothing to do with your discipline. If you just go and search some software, you will find there's so many softwares and programs available just for organic chemists. AI has been integrated everywhere. Right? So use that AI knowledge, is available in your field of expertise. Do not think that, man, I do chromatography, how can I use AI in chromatography? There is. You would be surprised to see how AI has been integrated into everything, right? And for AI, you just need a computer and internet connection. Develop those skill sets, learn as much as you can, and have your own companies. Make your own money. Okay, so this is the basic, all the docking that I mentioned. Now, how to do the docking experiment? How to do the docking using Chimera. It's very simple and easy. What you have to do, the very first thing, you already have downloaded Chimera so far. You need to download the AutoDock also. AutoDock Vina is the algorithm used by Chimera to do the docking. It is not built in Chimera. Right? When you download the Chimera, AutoDock is not downloaded automatically. You have to download it separately from a different site. What does it mean? It means that the Chimera can use a number of algorithms. Right? Here we are using AutoDock. We are asking you Chimera to use AutoDock to learn the docking. But depending on your need, you can use another algorithm. We are using AutoDock Vina. To download this AutoDock Vina from these sites, from Windows and Apple. And then step by step, we need to have a compound that you want to talk about protein against. So I'm using acetohydroxamic acid, which is AHA. The abbreviation is, and acetohydroxamic acid is a compound against urease enzyme, because I'm using urease protein as a model protein for the demonstration purpose, and the compound I'm using is the AHA. How to get yours, I will show you in the next slide. Then next, what you have to do, you need to have a canonical smile structure. That I will show you this on the next slides. Then once you decide your compound, you have to prepare your compound. I will show you this again. And then import your model structure of the protein from Swiss model, the one I mentioned in the beginning. You can import that structure, or if you want, you can just use a PDV structure of your protein, depending on what you decide, right? And then you have to prepare the protein, make a docking grid, run Autodoc Vina, and finally you will get the poses of the protein and the compound available to see which one is the best. You can choose which one you want based on the binding scores, right? Which is measured in kilocalories per mole. Okay, the very first thing is, okay, now to how to get the structure of your compound against your protein. You have you have taken one sequence. Now you know the structure of that protein, as I mentioned in the Swiss model. And now you want to know how many compounds have been reported against that protein or that sequence. So if you remember in the very first exercise I mentioned, go to UniProt website and then get from there you might have chosen a UniProt ID for one of your target proteins. That UniProt ID you need. To go to Campbell database, here in the Campbell database I write the UniProt ID, and based on that UniProt ID, Campbell tells you, okay, I find so these are the proteins, which one is yours? So you choose whichever protein is yours. Once you click on it and then you scroll down, you will find ligand efficiencies. These are all the compounds reported against your protein. There might be stimulators, inhibitors, agonists, antagonists, and each dot here, each dot is representing a compound. Right? You can click on each dot one by one and you will get all the terms of information about that compound against your protein, right? And if you look at their colors, they have different colors, red, magenta, and the colorless or something. So it is just telling you that what is the concentration at which your compound is active, right? So it is, for example, it's less than 1 millimolar, 1 is to 1000, 1/3. So here I think it says millimolars and the nanomolars and so, right? So here you can choose whatever compound you want. Which one I chose? I chose this one with the highest SEI index and the highest BI index. You can choose any compound you want, right? The AHA is the one, I use AHA as the standard inhibitor against my enzyme, and I do the same experiments in the lab. And I use this AHA here, chose it. When you click on it, see, this is the information about that compound. You can see the structure and all other features and the parameters reported about your compound. You click on them one by one, and then it will take you to that site on the web page, and you'll get more information about the same compound. You want to know how many derivatives of that compound have been made? You can get this information in here also. You want to know how many compounds have been established as drug? You can get this information. So these compounds everything has been reported about your protein. They could be drug. It could be only compound reported in the paper. There could be compound patented. There could be just reported initially at this screening stage or so. Whatever has been reported in different databases, that information will be. So you can choose any of your compound here, and that compound then you will use for docking studies. So what you have to do, very important thing is when you click on this any of the dots, here you see canonical smile. Canonical smile is the linear structure of your compound. It looks like this. So this compound, if I write the linear structure, it is written like this. Because the chimera designed the structure of compound by itself, so that's why they ask for canonical smile. So you can copy this for the chimera, right? And the other things that you can go and search, like click on this one by one. I don't want to go into much detail, otherwise it will be very boring. You can just go and explore yourself, okay? So here, the compound is, if you click on the substructure, this thing in here, structure search, this will open up in it's called editor also. Does it look similar to any software? This window here? Which one? CamDraw or CamWindow, right? CamDraw is for Apple and CamWindow is for the Microsoft Windows. Here, this is the structure of the compound in here, right? And if you want to know what similar structure of this compound has been reported, go to similarity. Substructures with different derivatives are reported here. And you can choose the threshold. Suppose I want 100% similar structures, right? I can make it 100 by using this. You see this green thing, you can move it left and right. I can make it 100%, I can make it 90%, I can make it 60%, 10%. So whatever similar structure to that compound has been reported, I can go and get those structures. One thing. Second thing, and which is very important, why it is opening in this editor. So this is a structure reported already in literature. If I'm working with this, what's the big deal? I'm just repeating something that's already been done, right? I want to do something new, something novel. You can change the structure of this compound. Anywhere. You can replace the hydroxyl group with benzene or something. You can replace the amino group with an alkyl chain or something, right? So you can design, make it a novel compound that never existed before, right? And that compound structure then you can use in docking. So that's the purpose of editing here. You can edit this structure yourself. Some of you, I think everybody knows about how to use CamDraw and CamWindow. Everybody knows CamDraw and CamWindow. Have you heard the name CamDraw? Yes, but not everybody, right? CamDraw is a software, or it's a software that just looks like this. When you open the software, it has these tools available on left and right. This is for making a single bond, right? This is for making a hydrocarbon chain. This is for giving a charge to an atom. This is for double bond, right? This is if you want to add an alkyl chain or R chain to your compound. Similarly here, these are the atoms available. So you can replace these groups with any of the atoms. So you can design your own structure and then save it. There are different formats available. So, STF format, PDB format, and other formats available. Sometimes I think JPEG images also. So you can save them and then use this structure for docking. So now you can design your own compound, a novel compound that never existed before, right? So with only few steps, see a project, a research project is ready for you. Okay, so that's all about the compound. So if you want to know the compound active against your protein, you can either get the existing one or you can design your own, right? So once whatever you decide, now come back to Chimera. In Chimera, as I said, you have to do certain steps. The first thing is that you make a docking folder. Make a docking folder on the desktop somewhere because you will be saving all the files once you've done docking in that folder. So if you save that folder, make a folder, then it's easy for you to go and retrieve the files. Next step is you have to make the structure of your compound in Chimera, and for this you have to go to follow the tools that I mentioned in here. Go to tools, structure editing. Where is the structure editing? Here. Tools, structure editing, build structure. A pop-up like this will open, and here you copy your canonical smile structure. Once you copy the structure, apply, and the Chimera will make the structure full, right? So rather, because Chimera I think does not import these structures in this form, it draws its own structure. So when you put the canonical smile structure there, it is made, and this is the structure that you need, right? So once you design you have the folder, have the structure in here, it's called ligand preparation, and then follow these tools. You need to prepare the ligand also, make it a little flexible. You need to orient it a little bit so that it can fix on the protein, and this is taken care by the software. But you just need to follow these steps. You should not take more than few seconds of your time. Okay, so this is how you've done the ligand preparation. Ligand is done. Now come to the protein. Again, for the protein, you have two options available. Either you can use the target protein, the PDB structure from the PDB database. You can get it in the PDB format, download that structure, and I think I showed in one of the steps in the previous exercises. Download the structure, or you don't want to use that. You want to use your 50 amino acids. Right? The 50 amino acid, the structure that you made from the Swiss model server, that is structure you wanna see that if you any drug you can find against those 50 amino acids. Here I'm using my 50 amino acid structure in here, right? So once you choose that, then you have to prepare the structure. If you're using structure from the PDB, even the one you use from PDB, and I think I mentioned to somebody that PDB structures are structurally, experimentally determined structure. They have used X-ray crystallography to make the structures. They have used NMR to make the structures. They have used cryo-electron microscopy to make the structure. The structure that you see in the PDB database, they are experimentally determined structure. They have invested amount, time, and labor to find out those structures. In that case, if you are using X-ray crystallography, and if any of you have done it, the X-ray crystallography gives you structure in so much detail. right? So much detail, sometimes it becomes very complicated to draw the structure based on the density map that you get. So what they do for analysis purpose, they remove all the hydrogen atoms from the protein structure, right? So the structure that you see in PDB database, X-ray determinant structure, they don't have hydrogen atoms in there. But when you're doing docking and you're using a structure from the PDB, you have to put those hydrogen atoms back, right? So this is what I mean when I say protein preparation. If you're using a protein, you need to go and give certain commands, follow these commands in tools, surface binding analysis, dock prep, and many pop-ups will open. Then you have to just press OK, 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 OK. This means you are putting the hydrogen atoms back, you're putting the charges back, bringing the protein into its native conformation, right? So once it is done, the third step is making a grid on your protein. What grid means? Why we need grid? Any idea? You see this green box is called grid.
To find the active site.
To find what?
Active site.
Yes. So if you already know where the active site should be on your protein and you know the size of the grid, you can play with it here. You know the numbers here you write. In the real time, you will see when you write numbers here, the size of this grid box is changed. So you can increase the size or decrease the size. In case you don't know where the active site is, because the new protein, novel protein, you have no idea, and you just want to do the docking done, then in that case we make a huge grid, right? Then we are asking the software to find out yourself. We have no idea, right? So in that case, again, you can choose the size by manipulating these numbers. So manipulate these numbers. So this is called the grid making. So what happens as the last step before you get the results? This is the fourth step. So now we are going to the tools, surface binding analysis, autodock VINA. When you click it, this thing will open. So once you specify all the parameters here, you click OK and the docking will start, right? So what is in there that you need to specify? For example, you have to let them know where you want the output file to be stored, right? So you just browse it and let them know I want it to be saved in my docking folder. Here you specify your docking grid, and then here there are many other things, like that you need to specify the receptor options and so on. So for example, here it is usually shown as false. You have to click the true. I mentioned things in here, right? And then you have to also mention how many poses you want. So if you want, the system can make 100 poses for you. It can put the drug at 100 places in the protein and say that, okay, you go and choose it, which one you think is the best. But the best thing is, like, I choose 9 poses. I'm asking them, just show me the best 9 poses. And then I will choose from those poses, right? And the last, you have to specify them which algorithm to be used. You are telling Alchimera which algorithm to be used. We want it to use autodock VINA. Autodock VINA, you have 2 options. Either you say, no, I don't want to download autodock VINA in my computer, and I want Alchimera to use the global autodock VINA, globally available somewhere, and use it from there. Or you can specify the one which is stored in your computer. The problem with the global one, the open web service available, it will take forever because it's too crowded. The entire world is using that globally available autodock VINA, so it will take you too much time to get your docking done. If you just use the one locally available on your computer, specify, browse it and let it know. And for all of you guys in your computers, autodock VINA, you have to specify this path. It is saved in your PC, then go to C drive, then go to program files, and then go to Scripps Research Institute, and then VINA. Right? So the VINA is made available and kept updated by the scientists at the Scripps Research Institute, and this is how it is downloaded with this path length and your computer. Just specify that path and then click OK. When you done click OK, you have specified everything. You have done everything. You have given the structure of your compound to the chimera. You have given the structure of your protein to chimera. You have done the ligand preparation, you have done the protein preparation, you have told it where is the grid, you already have explained all the parameters, you have mentioned how many poses you want, right? And you click OK, now the computer in the background is busy doing docking for you. For me, it takes only a few seconds. Sometimes I remember students have reported to me telling me, man, just the whole night is working, I'm not getting any. It depends, I think, on the speed of the computer and the internet. So usually if everything is ideal, it should not take more than a few seconds. Maybe at the most, maybe 1 or 2 minutes, but not more than that. So while the system is busy doing docking and finally displays the results, these are my results. So I have shown you the pictures of only 2. So here are 9 poses. 1, 2, 3, 4, 5, 6, 7, 8, 9 with their binding scores. So based on the binding scores, if you click on each pose, it will show you the picture how it looks like, right? And then you can see that there's one place it is showing to bind here, whatever ways the computer think. It is okay. Again, as I sometimes not always think that the AI is going to take your jobs. Right? People say now the AI is everywhere. Now those jobs going to be disappear, this and that. Here, computer doesn't know anything. Which pose is the best? Still, you are the final decision maker. You have to choose which one you think that is the right pose and the protein. Right? So the exactly same thing with the AI. AI, what you tell them, they will do. They are dependent on you. So nobody is going to take anybody's job. If you keep yourself smart and knowledgeable more than the AI always needs you. Right? So here the system is telling you, okay, I found these ones. Which one you think is the best one? So which one you think? These are the binding scores. The free energy scores here, the one that you're seeing. Which one you think is the most stable pose? Out of these 9s. Minus 2.8 is the most stable one? How many agree? Only 2 people? The word the rest think. Which pose is the most stable pose? Based on the binding energies mentioned here.
I think minus 2.5.
Minus 2.5?
Or maybe minus 2.
So the only criteria you have available right now based on binding energies, which one you think is the most stable pose? Minus 2.8? Huh? Which one is it?
Minus 2.8.
Minus 2.8? Minus Dr. Razuddin's scholar student is saying is minus 2.8. How many agree? Only see how many students they agree with you? Very few. And very tiny hands are just. What other students think? But I think I hinted somewhere about the energy that I asked you if the system, the nature requires huge amount of energy to maintain a structure. Is it stable or not? No. Then what, according to this criteria, which pose should be the most stable? With highest binding energy or with the lowest binding energy? With the lowest binding energy. So that pose is the most stable one because you don't have to do anything to maintain it, right? So the most stable is minus 2.8, right? Because it is the lowest energy. Minus 2.8 is lowest energy or not? Yes. Or some of you think is highest? Zahid, you understand? Minus 2.8 is lower or minus 2.2 is lower? Minus 2.8 is lower. Minus 2.8 is the lowest energy. Everybody agrees?
Yes.
Yes or no?
Yes.
I might be wrong. No?
Yes.
So minus 2.8 is the lowest energy, right? Any question about it?
Ma'am?
Yes.
But there are 2 different RMST values. For first minus 2.8, it's 0 RMST value, whereas for second one minus 2.8, it's different RMST value.
For this one, the second one?
The first and second.
In here, so that this is the reason we focus more mainly on the score, binding the score. But you will notice some of the students, they come back to me, they say, ma'am, I don't see any binding score. But in that case, they just see only the RMSD in one column. So then you decide based on the RMSD score. And what is RMSD is, I don't want to go into too much detail, it will become very boring. So once you are very much into docking studies, there are other, tiny details, technical details you need to learn and develop your expertise in. At that time, you learn about the RMSD. So I did not go in that detail, right? Because it's not too focused, too detailed understanding I want to give you. I just want to give you briefly know-how about how things work in docking. Any other question that you have? Something extraordinary or unusual that you see from these pictures?
One question.
Yes.
Regarding ligand preparation.
Okay.
When we run a ligand on a camera, is it possible to use the same structures from camera 3D as a docking?
Yes, you can. Who is stopping you? As long as you know the canonical smile, the structure of your, and it is easy to find out, right? Canonical is a linear structure of any compound.
Sometimes you say that to minimize first the ligand and then we use.
Yeah, this is called ligand preparation. You have to do it because then it just orients the structure in a way that it can be docked on the protein.
And use some
Software, you will see immediately. When you are doing the ligand preparation here, So when you carry out all these steps, you will notice, everybody will notice it, whatever compound you choose, and when you've done these steps, you will see immediately the structure is changed a little bit. Immediately when you're done and click OK, you will find that the structure of that compound a little bit changed. Orientation is changed a little bit. Why? Because the system is preparing the compound to be able to dock on the protein. Anything that you guys notice here in these structures? Huh? Okay, what's wrong with it?
Binding site.
Binding site
Huh?
is different.
Binding site.
Binding site is different?
Yes.
That's what you notice? So you guys notice that here the compound is not exactly superimposed on the protein structure. And which is this protein? These are my 50 amino acid sequence. My 50 amino acid sequence, I use that when I did not use the structure from PTP. If I would have used the structure from PTP, maybe the compound is exactly superimposed on it. And what is the reason? Why is there a distance between the compound and the protein? Idea? Any idea? Why there is a distance between the compound and the protein? It's not exactly superimposed. It has to be superimposed on the protein.
Box selection sometime.
Box selection, interaction, what else?
No compatibility, not strong compatibility.
No compatibility. Anything else?
No binding site. No binding site.
No binding site. So the thing is, the 50 amino acid that I've chosen, it's not necessary that those 50 amino acids have the active site or the binding site. Because if you remember the structures where I superimpose on the original structure in here, in the very first few slides, where is it? Here. So you see, this is the target protein, the entire target protein, and these are my 50 amino acid sequence, which is some sort of side loop of that original protein structure. The active site is somewhere here, right? The active site is here, and if you are trying to dock this protein with the compound, definitely it's not going to bind to it. But system doesn't know because he asked it to dock. It has done the docking and showing you the poses. It's now you have to decide. Oh, there's no binding site in there, right? So in case if you don't know the binding site, the best thing is just make a grid for the entire protein. First, once you find out the active site is in here, then do the docking experiment, right? So it saves you a lot of time doing docking with a protein sequence that doesn't have an active site or binding site. Get it? So there's so much detail and things that you can learn while you're working on it. But whatever you have learned so far should be enough for you to complete these experiments. The last lab report. I think that was the last slide I have. Anything else I have after this? Yes, and with this we've done our treasure hunting scheme, identify a protein from its unknown sequence. So initially when you started this course, the very first thing, the only thing you knew was the 50 amino acid sequence. But from that 50 amino acid sequence, we ended up having a drug against the protein, right? So these are the exercises we did, some of the steps we did, some of we don't. Successfully identified the unknown protein as well as its ligand, right? In just 3 different exercises. And what we do, usually I ask my students, so as I said, it's a complete project, it's a paper, it's a research presentation, and we have the opportunity over there at the University of Denver for the students. They come and present at the You Will Discover conference. We have a graduate student conference also where they can present, and they can publish it in different journals also. There are student journals. So you don't need your PIs to publish anything. I talk to many people, they say, man, my teacher is not letting me publish and so on. So you can do these projects on the side. Nobody is stopping you. Write your papers, write these papers, and then get them published. And when you get an opportunity, I'm sure in Pakistan also there are so many now conferences and workshops and research days happen where you can go and present your stuff. It's going to make your CV happy. Everything will come in your CV at the end of the day. You can mention them in the resume or your CV. So these are the journals that I recommend to the students where students can publish. And it doesn't need to be student from the University of India. It could be from anywhere. You guys also can publish these journals. There are tons of journals. When we were studying, I didn't know any grant where I can apply as a student. Here I find out there are so many grants you can apply as PhD student, as postdoc, as research associate. You don't need to be a PI to get access to those resources, right? Similarly, you can, there are many journals where you can publish as a student in your capacity. For a student, the criteria is a little milder. It's easier for you guys to publish as compared to a professor. So there are different journals, publish this work. So I share this with my students, and that's it. The research project is concluded. Right? To see that within these 3 exercises, we learn how to work on a project and do the research, come up with your own creative ideas, write the paper, and so many things. Any questions? What time is it? 1:30? We finished in 1.5 hours. Right? So this is today's last class. I don't know if anybody is missing the class. Perhaps I think they have the. I don't know. Did you record or you did not record? It was recording. It was not recording. So I think maybe these 2 lines they will hear only the recording. Recording is not recording. Maybe the ones who are not available, you can let them know. They can have access to the Google Drive, the recording. This one I will upload. I thought I did last time, but maybe I forgot. So I will upload these ones, right? But the last lab report is you have to just follow the questions that I